All right. First of all, I would like to thank Andrew and everyone else for inviting me today. It's a pleasure to talk about my favorite topic, and I find it quite exciting to talk about elemental stoichiometry in an auditorium named after Redfield. All right, before I start, I just wanted to acknowledge that this is very much a collaborative project among many people, especially at UCIrvine, where I'm stayed. A lot of people, both modelers and experimentalists, have been involved. We also have collaborations at Princeton with Simon Levin's group and his excellent postdoc, George Hagstrom, and Mike Lomas from Bigelow, and I have gone on multiple cruises. And then lastly, the people from the Go Ship program, Lynn Challey et al., that have helped us get on many really nice cruises around the world. And last but not least, NSF, especially biological oceanography, who has funded a lot of this work. All right, so I probably don't need to spend too much time on the Redfield ratio in the Redfield auditorium, but nevertheless, I think this is a fundamental property of biological oceanography. And if you, for example, read the book by Sarmiento and Gruber, Ocean Biogeochemical Dynamics, you realize that pretty much on every page, this ratio creeps in to the concepts that we base a lot of biological oceanography upon. And because it forms this very tight link between nutrient limitation and the potential for carbon fixation and carbon storage in the ocean. So having a static relationship between how much carbon and how much nitrogen and phosphorus you can have in a cell forms this very static link that really propagates throughout so many concepts between how what is the role of nutrients and what then becomes the controls on carbon fixation in the ocean. So really you could say that this concept has an immense influence on ocean biogeochemistry. Likewise, as you start to maybe acknowledge that maybe this ratio is not quite as constant as that, that really starts to move some fundamental concepts in oceanography. And I'm going to talk just about one, which is about carbon export and how we can use our knowledge about phytoplankton physiology to understand regulation of carbon export. But I could give a very similar talk about nitrogen fixation, for example, and some similar ideas would come up through that. And so I cannot emphasize how important it is to understand this ratio in biological oceanography. All right, so some work we did now about 5 years ago. We did a meta-analysis where we compiled all, actually the few, measurements of particular organic carbon, nitrogen, and phosphorus. And especially at that time, particular organic phosphorus was a rarely measured property. And we got about data from about 1,000 places around the world with a heavy bias towards the northern hemisphere. And the first pattern that started to appear out of that is that the elemental stoichiometry of the ocean and the CDN to P ratio of phytoplankton communities are not constant. In fact, it seems to be rather variable, where we see that, for example, here represented by the C to P ratio, that it's rather low in high latitude environments, often substantially below Redfield proportions. However, as we get into lower latitude regions, we see ratios that are substantially above Redfield proportions, especially in the oligotrophic gyres. And then in the last sort of major biome in the ocean, the equatorial upwelling zones and coastal upwelling zones, we see that the C/P ratio is probably close to Redfield proportions. However, this is like a meta-analysis, so it's like all kinds of diverse samples taken from all over the world, taken with different methods, etc. So one of the first things that we wanted to do was to get a much better data set on understanding the distribution and the elemental composition of particular organic matter in the ocean. And so we've gone on a number of long cruises around the world, and this is still very much in progress, to all the major ocean basins and basically measured particular organic matter. Today I'm going to give you one example from the Pacific Ocean, from the P18 cruise from La Jolla all the way down to the ice edge of Antarctica. But we are accumulating data from all these different places to understand what is the field distribution of particulate organic matter and the elemental ratios of that. And so here is the example from the P18 cruise. And what we see is that we can sort of see the different biomes just in the concentration of particulate organic carbon. So as we would expect, particulate organic carbon is lowest in the two ocean gyres, so up here in the north and down here in the South Pacific Ocean gyre. It's elevated near the eastern equatorial upwelling zone, and then it becomes rather high as we move into the Southern Ocean. We see a similar pattern in the particulate organic phosphorus, but since today is about elemental ratios, I wanted to show you what the C to P ratio is. So both in the North Pacific subtropical gyre and in the South Pacific subtropical gyre, we see ratios that are substantially above Redfield proportions, whereas it's close to Redfield proportions, but perhaps a little bit above in the equatorial upwelling zone, and then it drops down to rather low elemental ratios as we get into the Southern Ocean. So if we summarize that in terms of different ocean regions, we can see that the highest ratios here, right, is in the South Pacific subtropical gyre, and it can get up to about 250, but the mean is somewhere 170, 180. It's also slightly elevated in the North Pacific tropical gyre, although we didn't get quite far west enough to really see low nutrient concentrations. I would expect the ratio then to be somewhat higher. Again, it's sort of depressed, but maybe not quite at Redfield proportions in the equatorial zone. And then we see within the Southern Ocean that there are sort of different regions defined by different fronts. And we see that progressively that the elemental ratio drops as we get towards the ice edge. And so within the Southern Ocean, there also seems to be sort of different water bodies with unique elemental ratios. And this is something that we're trying to understand right now. So after we've sort of mapped out from a variety of cruises, and I just gave you one example here, the next question, of course, that we want to ask is what is the underlying driver for the variation in C to N to P? And one of the problems that we're faced with is that there's a very strong covariance between multiple environmental drivers in the ocean as we go from sort of high latitude towards low latitude environments. So we go from low temperature to high temperature. We go from high nutrients to low nutrients. And we also have shifts in the phytoplankton community composition. So we have this sort of latitudinal pattern with low and high latitude waters, high in gyres, and maybe intermediate at the equatorial upwelling zones. And now we then want to ask, what is sort of like the underlying drivers for this variation? And the first set of experiments that I wanted to show today is where we tested basically what is the effect of nutrient limitation, and particularly nitrogen and phosphorus limitation, on the elemental composition of Synechococcus. And in general, we would expect that when cells are nutrient limited, they would have a low cell quota of that particular nutrient, and thereby a high C to P or C to N ratio, and vice versa when that nutrient is replete. However, what we've also learned from, for example, Richard Geider's work and others is that there's also a strong interaction with how fast a cell is growing, meaning that when you're growing really, really fast, maybe you have less flexibility in your elemental composition compared to when you're slow. So we would also expect that there would be some kind of interaction with the overall growth physiology in addition to nutrient limitation. So to achieve this, we did some chemostat experiments, and I just want to say that these are really complex experiments. They may seem simple, but 3 years of work of my postdoc went into this, and which was sort of, at least to me, a cautionary tale of how many different environmental factors could you manipulate for this because several years for each factor. You can easily account for several years per factor in this type of experimental setup. But basically, we're manipulating, we're going from a nitrogen-limited condition where the N-to-P ratio in the input media is 6:1, and a P-limited condition where it's 70:1, and then we manipulate it with 4 different growth rates. All right, so as would be expected from, for example, Droop model, we see that as a cell is growing faster, it has a higher cell quota of, in this case, of phosphorus. All right, so this is sort of classical phytoplankton physiology. Also, that the cell quota is lower under P limitation than it is under nitrogen limitation. So really no surprises in this. When we look at N limitation, we see a much smaller effect in Synechococcus. Actually, N limitation had a very small effect on the N quota. And to me, that suggests that there's low potential for nitrogen storage in Synechococcus at least. But again, we also see sort of the general support for a Droop-like model. But the surprise that we saw in this experiment was with carbon. In reality, it probably shouldn't have been surprised because I think that many experiments in microbiology have shown that the faster cell is growing, the larger it is. But somehow I have forgotten that and realized, and so when Nathan Garcia, the postdoc in my lab, did this experiment, what we also saw that the faster they grow, the larger the cells are and the more the carbon they have. And this is an important component because typically when we do phytoplankton elemental stoichiometry models, we tend to assume that the carbon quota is constant, and what we need to keep track on are the variations in the P and N quotas. But what we found here was that the C quota also was highly dynamic, and that made understanding the overall elemental ratios somewhat more complex. So if we look at the elemental ratios, what we see is that P stress is really important for changing the C to P ratio, whereby when cells are growing slow, Synechococcus can achieve like a 300:1 C to P, but then when they're approaching the maximum growth rate, the C to P goes down quite a bit. Whereas we saw very little effect of N limitation across different growth rates. Basically, when they are N limited, N to P and C to P ratio was largely constant. So what we found, at least with small phytoplankton, is that the key to understand the elemental composition of a small phytoplankton like Synechococcus is P stress and the degree of P stress. All right, so the next thing we wanted to do now was to build this information both from the field data and from the lab data into a trait-based model. And this is work led by George Hagstrom at Princeton. And so we wanted to design a trait-based model that is based on the optimality principle, as Richard Guyder talked about before. And essentially, it sort of aggregates both acclimation and adaptation into one process. And then what I'll show you is how we embedded this trait-based model into an ocean GCM to understand, with an optimized P cycle, to understand carbon export. All right. So just to give you a brief rundown on our trait-based models, and it's really sort of building upon a long tradition of phytoplankton physiology models, and ours in particular was inspired by the Danes et al. Paper from 2014. We basically have 4 components: a structural component, a light harvesting component, a biosynthesis component, which are really ribosomes, and then a storage component, which is by and large polyphosphates. And then over sort of different growth rates and different cell sizes, we predict different allocations to these different components. We have certain environmental inputs, and then we look for what is the optimal growth rate at each environmental condition, and that then gives us the predicted C to P under those particular environmental inputs. And so we're basically assuming that a phytoplankton that grows in a particular environment is like the fastest growing and is optimally allocated and in complete equilibrium with the environment. This is obviously an assumption that I'm sure there are many environments where such an assumption might fail, hopefully it won't fail too much. All right, so the other thing we did was that we optimized the model against our global particulate organic matter database. And we were interested in figuring out which parameters, and there are 14 parameters in our model, which of those 14 parameters are the ones that gives the highest sensitivity in the model, and then trying to find the parameter values that best predicts the global distribution in the POM concentrations and ratios. And what we find is that the model and the C to P ratio of phytoplankton is particularly sensitive to how we parameterize a P storage. This was also a conclusion from the Danes et al. Paper, and so there seems to be some consistency in that. Unfortunately, we don't really know very well what regulates P storage in phytoplankton, so that is a major problem. But nevertheless, that parameter that controls the P storage is by far the most important parameter, and one that we really need to understand in the laboratory in order to understand what controls the C to P ratio of phytoplankton. Another one is the allocation, how much of the biomass is allocated to ribosomes, and then finally the ribosome T's temperature sensitivity. We then optimized the model against the POM data set, and then an outcome then is that we predict the highest C to P ratios in really low phosphate, so below 10 nanomolar, which again is a problem that we don't measure phosphate at those levels that well. But nevertheless, really low phosphate values are important and really drive C to P, but also high temperature. So there's both a strong temperature and a strong phosphate effect, whereas perhaps a less effect of light levels. All right, so now we embedded this trait-based model into a global ocean biogeochemical model. And then in the ocean biogeochemical models, we have information of light, temperature, and phosphate levels. And we can put that into the trait-based models and then predict what is the C to P of exported material. And I compared this to a very simple empirical model we published a few years ago, which are purely calibrated just on phosphate. And there are some differences, but also a lot of similarities just between a pure phosphate-based model and then this more complex trait-based model. And that is because the P storage and the impact of phosphorus stress is an important component. But nevertheless, we see replicate sort of the ideas from before with high ratios in the subtropical gyres and lower towards high latitudes and sort of intermediate levels in the upwelling zones. All right, so then we could also predict the carbon export flux out of this GCM. And this is where we can start to influence maybe our understanding of global ocean biogeochemistry. For example, in the Redfield model and in our general concepts, we sort of tend to think that most of the carbon export occurs in places like the Southern Ocean and equatorial upwelling zones, etc. But what we see is with the trait-based model, because we see much higher C to P ratios in the gyres, that pattern shifts. And really at the edges of the upwelling zone, where you still have a somewhat strong nutrient flux, but the phytoplankton drives down the phosphate to low levels, it becomes the regions where we see the highest carbon export flux. So for example, in regions like here, now becomes real hot spots for carbon export. So we see quite a strong shift in where we see the most export. And that's just depicted here by the comparison between the trait-based models and Redfield. And we see that it's 2 to 3 times as more carbon export when we use this trait-based model in oligotrophic gyres. So by having a more realistic description of the C to P, we start to see rearrangements in where the carbon export would occur. Now we can say, is that correct? Is it real? And so to test that, we compared our carbon export with the Redfield model, this phosphate-based model, and then this trait-based models against global trap data. And so Colleen Mao had luckily compiled a lot of trap data, and so we used that data, and then we calculated basically what is the bias between our carbon export compared to the trap data. So if you have a strong positive bias, for example here, that means that the export model overestimates compared to the trap data, whereas if you have a strong negative bias, then that means that your model is underestimating the export. And I just want to bring your attention to two parts here, which is the Southern Ocean and the North Atlantic Gyre. In the North Atlantic Gyre, that's where we predict the highest CDP because of the severe P stress. And what we see is that in the Redfield model, it severely underestimates the amount of carbon export compared to the traps. Whereas both in the trait-based model and the Gilbert-Martini, this phosphate model, this bias largely disappears. In contrast, the Southern Ocean is where we have the lowest CDP ratios predicted with a trait-based model. And what we can see is that the Redfield-based model vastly overestimates carbon export in the Southern Ocean, whereas that bias disappears with either the phosphate-based model or the trait-based model. And so the regions with the highest CDP and the lowest CDP, you remove a lot of the bias between the export and the trap by using a variable stoichiometry. And thereby we get a more accurate description of carbon export by using variable stoichiometry. So to conclude, I wanted to say that we see clear latitudinal patterns in CETA and P, both in this sort of global meta-analysis and as well as our individual cruises. We see that culture experiments, and I just showed you one, but we've done a whole series of them, can help describe biological mechanisms regulating CETA and P. We can use trait-based models to bring in all that knowledge and optimize them against the description of elemental stoichiometry as we see in the field. And we now can embed that into global biogeochemical models and find less bias and a more accurate description of carbon export. And in particular, that maybe we have traditionally underestimated how much export occurs out of the gyre. And with that, I would like to thank you for your attention and take any questions.
Hi, Adam. Thanks for the talk. Is there any work that you and your group are doing to extend these changes in CETA and P towards oxygen consumption? So looking at how, if you change the physiology of a cell based on your CETA and P, then surely that extends to how much oxygen is required to remineralize that organic matter.
Yes, that's an awesome question. Very much indeed, that's the direction we're trying to push it right now. You should say it really loud to NSF as we're trying to get a proposal funded for this work. But yes, I think that's a really interesting thing. It doesn't have so much to do with the nutrients, but more the composition of carbon and the amount of hydrogen and oxygen there is. So how oxidized the carbon is in the biomass. And in our preliminary data, there's indeed quite a bit of differences in the oxygen demand to carbon, so to speak, oxidative demand. But that's still very much new data, and I can't yet say if there's a systematic pattern or not, but I think that's exactly that. I think also pushing it towards the micronutrients, that's another direction. So these two directions are ones that we definitely want to include. Great question.
During the Southern Ocean JGOF study, one of the things that was observed is that, consistent with the C to P ratios you described, there is preferential uptake of phosphorus during the growth phase of the bloom, and so that would lead to low C to P ratios. That was attributed by Stephanie Rubin in her thesis papers to phosphorus storage by diatoms. But what Stephanie also noticed is that by the end of the season, before the organic matter was all exported, a lot of that extra phosphorus had been regenerated. So perhaps the export C to P ratio is more like Redfield than the low ratios you would get by filtering healthy diatoms out of the water column. At least, so that may bias your high latitude. If that's really what happened, it may bias your trait-based model carbon export at high latitudes. I don't see how it would bias the subtropical gyres, but I wonder if you've looked into that at all.
Yes, we have indeed looked into that. That is definitely a comment that multiple people have made about, so to what extent is the variation that we see in suspended particles in the surface translated into exported material? And I think that's a really valid and important aspect that one needs to understand. And we've addressed that in a couple of different ways. One is that there's a few trap-based studies where they have measured other nutrients, like particularly organic nitrogen and phosphorus, for example, hot and fats. And we see that the elemental ratio of the suspended particles are very close to the elemental ratio of the traps. Actually, sometimes the CDP of the traps is a little higher than the CDP of the suspended particles because of preferential Riemann of P. The second way that we address that question is using inverse models, where we have basically the global distribution of phosphorus, phosphate, nitrate, and CO2, and try to say what is the elemental composition of exported material that best match the hydrographic distribution of phosphate, nitrate, and CO2, or DIC, I should say. And what we see is that the patterns in the suspended material and the predicted elemental ratios of the exported material through these inverse model analyses are extremely closely related. That we also see quite low predicted CDP for example, from the Southern Ocean using the inverse models. So very similar to what we would expect both from the POM and the trait-based models. So I think there is a larger convergence of what we see on the phytoplankton labs, what we see in the field-based measurements, and what we see in the inverse models that all sort of points towards the same patterns on the CDP. So in that way, I would argue that that it's not just that the suspended material is unique compared to what gets exported, but they're actually quite closely linked.